And okay. Okay, so technically speaking, yeah, this lecture will be our last lecture. So as I mentioned, because we spend a lot of time uh, studying the very fundamental important theorem, but now from lecture from chapter 13 and on, so we're going to move very quickly. So those are all about the application. So before we started before we start the lecture, so let's uh, briefly discuss the schedule. Okay, so let me start this. All right, so the term paper is due tonight. So any problem? So do you need more time? Yeah.
I thought it was due this morning.
Uh, 12, I think it's 11, 59 p.m., right? So that's tonight. Don't worry. So you can take it back and revise it. No problem. So in case you need more time, let me know. So I think, yeah, I can extend this to probably to tomorrow. Uh, this is for the term paper. And if you have any question about your term paper, so let me know so I can help you before the deadline. And so this weekend I have made a practice test. So I've already uploaded it to the iLearn. So yeah, please take time doing some practice on that. So that's the standard. You're gonna see the problems in the real final exam. And so our plan for Thursday is, so in the morning, we will have the group presentation. So each student will have like 10 minutes. So 10 minutes for your presentation. So just prepare a couple of slides, just like my lecture note. So and to summarize your term paper. And after the 10 minutes presentation, so we will have a 2 to 3 minutes question and answer session. So this is the place where you can earn extra credit. So don't just focus on your own presentation. So after you are done, so please pay attention to other people's talk. And so yeah, that's the place you can earn some extra credit. So the term paper we have put 50 points on the term no for the final exam we have 50 points. So another 50 points will come from the combination of your term paper and the presentation. And if you can ask questions and answer other people's questions during the presentation, so you can earn some extra. Yeah. All right, any questions? All right, perfect. So let's get started. All right, so I'm gonna stop video and okay, so today we're gonna chapter 13. So here we're gonna learn random model. So what is random model? Because in the past the regression model we used for testing either the QTL or a SDL. So we use a fixed model. So in the past, so in the previous chapters, we learned the regression model. Those are in the fixed model category compared to here, the random model. So why is that? Because so in the previous chapter, so we only deal with a controlled population. So the population, study population, mapping population is under the is under our controlled population. So what is controlled population? Controlled population. So either it's a backcross, right? Or it's a F2, or if it is a bit more complicated, so it's a four-way population. So in that case, in those cases, because the reason why we call those populations are the controlled population, because uh, at each locus there are very limited alleles. So for example, in backcross or F2 uh, population, at each locus there are only two alleles, right? Either A1 or A2. In the four way, so we may have like four alleles A1 from four founding parents, right? A2, A3, and A4. But in some studies, so if we do not have this control on the study population, the mapping population, so we do not know how many alleles could come up from your study population. For example, if you are studying a natural collection from uh, uh, a natural collection for a plant, for example, rice. All right, so there might be for each locus there might be many alleles. So in that case, or if your study population is a very complicated population, so we have to use a random model. So what's the difference between the fixed model and the random model? So in the fixed model, so I hope you still remember this term, the A, right, the additive effects, and D is dominant effects. All right, so in a linear regression model, like Y equals to mu plus Z, A plus W, D, and plus error. Okay. All right, so this is the linear regression model. And if it is a fixed model, so we call this effect, the additive effect, and the dominant effect are the fixed effect. So in this study, we will use our data to estimate this effect, right? So we use MLE, the maximum likelihood estimation, to calculate how big the magnitude of these effects. So because they are fixed, so we can estimate their effects. So that's why we call this fixed effects. But however, in the random model, because there's so many different alleles, so it's very hard to estimate the additive effects or the dominant effects for each different allele. So that's so if we want to do that, so then we will have too many parameters to estimate. So that's horrible. If we have a limited, if we have a very limited sample size, but if we have to analyze, but meanwhile, if we have to estimate too many parameters, genetic effects, so you know what's the consequence, right? So because there's no unique solution, because so your unknown parameters, the number of your unknown parameters are greater than the number of data points. So there will be no unique solution for each of them. So in this case, we have to use the random model. So instead of estimate each individual genetic effects for each allele, so we just estimate the variance of the genetic effects of the allele. So then the situation, oops, so either variance A or the variance dominant. Okay, so in this case, so we do not have to estimate too many parameters. We only need to estimate one parameters for additive effects at this locus, and another parameter the variance of the dominant effect at this locus. So you can think about this strategy as a dimension reduction strategy. So you, for example, if you use fixed model, you have to estimate probably 100. If there are 100 alleles at this locus, you have to estimate 200 parameters. But if you use random model, in this case, for this locus, you only need to estimate 2 parameters. One is the variance of the additive. The other one is the variance of the dominant effect. So is that clear? So that's the difference between the fixed model and the random model. Any questions?
Not quite clear. So
Oh, okay, go ahead.
the fixed, I'm just throwing out. So the fixed model. You know what? I have no clue. I'm not even
Okay.
going to try to
All
guess
right,
it.
Okay, got it. So probably, yeah, let me put this in another way. So, just got, okay. Okay. All right, so let's start from the model that we are very familiar with. So this is the model.
Mixed linear model.
Yeah, the linear model. And oops, this is additive, and this is the dominant. Okay. All right. So because we so in the past let's use the F2 as an example. So we have 2 alleles, right? A1 and A2. So here we try to estimate the value of this A and the value of this D, right? So if you so what is this value? This, so if you draw this, this is A1 A1, A2 A2, right? And so this is, let's say this is A1 A2, something like this, right? So the A is this, is that right? Or
Yes,
the A is,
I understand this.
yeah, okay, good. All right. So if you think about this, so this can be translated into the difference of something like the allelic effects, right? Something like that. So the little a is something like the difference between the two, the allelic effect between the first allele or the second allele, something like that, right?
Yes.
Okay, so all right, that's easy because we have 2. So suppose at this locus, think about a natural collection of the rice. So you have many different sorts of the parents, the ancestors. So you could have like maybe, let's say 10 alleles, right? And so on, right?
Mm-hmm.
So that's a problem. So you cannot use a single A to model your model. Is that right? Because all different alleles may have different allelic effects. So so the combination of 2 will make you crazy, right?
Yes.
Okay, so what we can do, so if we still want to use the fixed model to estimate something like this, so in this case, so in this simple case, we actually estimate the real effect from A1 and the real effect of A2, and the difference, something like the difference between these 2 will give you the little a value, right? Something like that. Yeah. So if you have like 10 allele, so if you still want to estimate the effect, real effects, how many values, parameters do you need to estimate? 10, at least 10, right?
Mm-hmm.
Okay. Then suppose you only have like 10 data points. So, and this is 10 parameters to estimate. Now think about the dominant effects, there are another set of parameters you need to estimate, right?
Mm-hmm.
Okay, so let's say you have 20, but if you only have 10 data points, 10 individuals in this population, so that's impossible for you to get the unique solution. Do you agree? Do
Yes.
you agree? Okay, so let's say if for each individual, the additive effects for for the A allele is little a1, little a2, all right, and so on, little a10, all right? Okay. So in fixed effects, you have to estimate them all. Then you have 10 parameters to estimate. However, in the random model, if we assume any of this AI, I equals to 1 to 10, is coming from a normal distribution with mean equal to 0 and a variance of A. Then, so if this is true, those are all the realized data from this distribution. So in this case, we only need to estimate this one parameter. So we use this strategy to successfully reduce the dimension of the problem. So instead of doing the estimation for 10 parameters, we just estimate this one parameters here.
Estimate one parameter so you can get close to estimating for all.
Yes, yes, yeah, you got it. Yeah, that's the key point. That's the key point. So that's why the random model here. So we are more worried about the estimation of the variance rather than the real value of each of these effects.
All
If
right.
we assume all the effects are a random sample from the same normal distribution.
Got it.
Mhm. Good question. Yeah, actually, this question is really hard. I mean, yeah. So in the a mixed model problem. So yeah, this is a very hard question. Yeah, point. Good. Yeah, I'm happy you asked this question. All right. Any other questions here? I hope it's clear. Yeah. So again, so in the past, we're dealing with very easy population, the controlled one, like by a Lilik system, right? At each locus, you only have like 2 alleles. Then that's easy. We can use the data to directly estimate the effects for each allele. But in this situation, so if you have a very complicated study population, if you have many alleles at each locus, then the fixed model is not appropriate. It's not appropriate. It's not proper. So then we use the random model to achieve the reduction, the dimension reduction, dimensionality reduction. All right.
I have a question, Professor.
Yeah. Go ahead.
So,
Yeah.
I'm not quite sure why for each locus there are more alleles. So this is for not for the diploid organism, or
It's still for the diploid organism. So if you think about the 4-way, so 4-way is what? So we start from the first parent.
yeah. So for the 4-way there is 4 alleles.
Yeah. So even for the 4-way is a control. Is a we still think about 4-way as a controlled easy population. If
Yeah.
you so if you do a very large study, like for humans, so um, if you study all the human in on this planet, so think about the locus. So I mean, not for all the loci, but for some locus, probably you may have many alleles, right?
Mm.
Yeah, that's the reason why. So why we cannot use the fixed model to do this analysis, because if there are too many alleles there, so rather than that, rather than using the fixed model, we use random model.
Mhm. Okay.
Mhm. Thanks. Yeah. Any other questions? Okay. All right. So when we use the random model, and a very useful study example is so when we want to do a study on the full SIB family. So full SIB means like all the sibling, all the progeny are from the same parents, father and mother, and this is a very important concept that we need to study, we need to learn. So the IBD, the identity by descent. All right, what does that mean? That means like so when we look at different alleles, so this term is relevant to the relationship between different alleles. All right, so the IBD is a special terminology in quantitative genetics to describe the relationship between any two alleles. So if these two alleles are the same copy, if these two alleles are the same copy from a ancestral allele in the past, then these two alleles are said to be identical by descent, so IBD. So in contrast, if so still we're talking about two alleles. If another two alleles are so they are of the same form, but we do not know if they are from the same origin. So then we say these two alleles are IBS, so identical by states, not by descent. So for example, in this diagram, as you can see, if you look at this two siblings from this full SIB family, and the first sibling, so the first A1 allele, and the first allele, A1 allele for the second sibling, so these two alleles are coming from the same from father. So these two alleles are said to be the IBD. They are the identical copy from the same ancestral allele here. However, if you just look at these three A1 alleles in these two siblings, so these three are called the IBS one another. Right. Okay, so if you compare this, so this design, this model is specifically for the full SIB family study. So if you compare this to SIB, the SIB1 and SIB2, there are 4 by 4, the 16 possible combinations, right? So for example, if both, so if in these two SIBs, in these two siblings, sorry, and both alleles are the IBD, then there are two IBD alleles. All right, so the diagonal entries represent this situation. And okay, if we use pi to represent the IBD value between these two siblings, so the expected IBD value will be calculated using this. So we are very familiar with this, right? So that's the probability, that's the one possible value for this pi, and that's the probability associated with this value, and we add up all three possibilities. So that's the expected value for the IBD value. And that's the variance, how we calculate the variance of the IBD value when we compare these two siblings from this full SIB family. All right, questions?
I have a question,
Yeah,
Professor.
Mm-hmm.
Where does the 0.5 come from for expected IBD?
Let me discuss this and then. You mean this 0.5?
Yeah.
Is this 0.5?
Yeah.
Okay. Okay, sure. So let's look at this one. So we have
Mhm.
1, 2, 3, 4. That's one.
One
That's
fourth.
4 over 16. That's 1 over 4, right?
Right.
Times
Okay.
this. So that's represented the IBD value equals to 1 because both alleles are IBD and the total 2 alleles pi equals to 1.
Oh.
Okay, so how about this? So this represent this 1, 2. So look at the 1 in the parentheses. So that means like 1 out of 2 allele are IBD. So how many of them?
Okay. Okay.
So
That's like 8.
yeah, that's 8
Out of
over
16.
16. But in this case, the pi equals to 1 allele out of 2 are IBD, right?
Okay.
Same thing for here. So if both are not IBD, so then yeah, but this term is gone, 0, right?
Hmm. Okay.
All right. Any other questions on this slide?
No.
Yes. Full
Oh yeah,
sibling.
go ahead. Yeah, right. Mhm.
So they're full siblings, and we have the expected value, which is pi. I don't understand the reason for the 1/2 and 1/4 values. Did we go over this before?
Oh yeah, sure.
Did we?
Let's go over this again. Sure, no problem. Okay, so let's look at here. So this is one SIB, right? This is another. So that's the first probability that the first SIB carried the identical alleles for the second allele, a second SIB. That means like at this. So both alleles are the IBD for these two SIBs, right? We put 2 here, so that's the 2.
And
so
then
in this situation, there are 2 IBD alleles, right? And this is the same for this one. That's why we see a 2 in the parentheses here, another 2 in the parentheses here, another 2. So out of 16, there are 4 possibilities for this kind of situation. So that's why 4 out of 16, which is 1 over 4. All right, so for the
okay.
yeah, so there are 8 such kind of situation.
But the value is 0.5 and 1.
Yeah, because we are talking about so for example for SIP1 or SIP2, there are 2 alleles, right?
Mhm.
So the first if both alleles are the IBD, here we use 2 represent. Then in this case, the IBD value should be equal to 1, which means 2 out of 2 are IBD. That's why this is 1.
Okay.
But if only one allele is the IBD, the other one is not, then the pi equals to 0.5 because it equals to 1 out of 2, right?
Yes.
That is, yeah. And a 0, you know what that means, right?
Exactly.
Okay, good. Any other questions here? Okay, let's move on. All right. So in case you do not have the clearly information about the status of the 2 alleles between these 2 SIBs, so then you have to estimate this pi, right? So in this, so for example, if at a unknown log, so you can if you do the QTL mapping, so you have to test not only the marker positions, right, but also you want to test any genomic loci on the genome, even between the markers. So for some loci between markers, you do not have to observe the genomic data, the genotype data. You have to infer that. So in that case, you have to estimate these IBD values at any unknown genomic loci. Is that clear? So we can use this to calculate the expected value for the IBD value when we analyze this full SIP family data. All right, and okay, so suppose we have two SIPs here, and we use very similar regression model, but here there's a little bit different between this model and then the model we used to use in our previous study. So here, the gamma the same. So suppose we are testing whether this locus is a QTL for the traits, and the gamma is used to represent the genetic effects, for example, the additive effects from this for this locus from different alleles. And so here, this A, so although we use A, please forget about the previous definitions when we use A. So in the past, we use A to represent the additive, but here, no, this A represented the polygenic effects. So what is polygenic effects? Suppose this is a genome, so there are many loci that are contributing to the phenotype. So when we do the QTL, suppose we are testing this position, this locus. So in order to achieve a very accurate test, so we in this regression model, we also need to consider the contribution from other loci. Do we? So we have to, right? All right. So when we use a linear regression model to test this locus, we also need to consider to include the joint effect from the rest of the genome that are also contributing to this phenotype. Is that correct? So this is why we use this model. Okay, so here this is this is the intercept. So don't worry about this. This is the error, and this is the genetic effect we are interested. We are interested in, and we want to test if for this locus, if this is equal to 0 or not, right? And this is the sum of the genetic effects from the rest of the genome when you exclude this genomic locus of interest. Is that clear?
So in the linear regression model, we don't account for these joint effects of loci, right?
Yeah, so in the past, because for the simplicity, so we do not, because in the previous chapter, so we try to introduce the very basic concepts to you without confusion. But now, from now on, so if you want to do advanced analysis, you always need to include these polygenic effects to make your model more accurate. Because when you do the test, you cannot just ignore the effects from the other loci, right? You cannot just test a single locus without considering the effects from other loci, right? Yeah, so this is how we use this polygenic genetic effects to capture the joint effect from other loci from the rest of the genome when you want to test this locus.
Okay,
Make sense?
got it. Yep.
Okay.
Thank you.
So these are this is the same equation, but for a different genetic loci that might have an effect on.
Yes.
Okay.
So, yeah. Okay. So for example, so why it's difficult to predict the stock market? Because if you always like include one major factor in your regression model, you cannot predict that the market, the trend of the market, stock market, right? Even you capture the major factor. So, but if you develop a prediction model, only including that single major factor in your prediction model, Can you get a very satisfactory prediction results? No. So you have to consider other factors, even they are not as big as even they do not they do not have that as big as the effect than the major factor. But it's better for you to include everything in your model to improve the prediction accuracy. Do you agree with me?
Yes.
Yes, so this is why here. So even you just want to test if this single locus has any genetic effects that can contribute to the phenotype of interest, but to increase the accuracy of the test, it's better for us to include other genetic effects in the same model as well. So that's why now in the future, when you do your own project, when you finish your data collection, if you use GWAS to analyze your data, so this is the model you need to use. You cannot use the previous simple model.
Mm.
Okay. So probably, yeah, when you work with Shibor in the future, so you're gonna listen, you're gonna hear about like the kinship matrix, the polygenic effects many often. Why? So that's related to this term. So polygenic, and when we talk about the polygenic effects, so we need to consider the relationship between the individuals in this study population, right? And we use this structure, we use this polygenic effects and the variance structure of this term to capture the kinship, the relationship between each individuals in this study sample, in this study population. All right, so now we are okay with this advanced linear model here, right? And so if we want to analyze these two SIPs simultaneously, we use this matrix. We put these two individual in this vector, and this is the linear model using the matrix notation, and this is the expected value because all this are the random effects and the average of them assumed to be 0. So that's why if you calculate the expectation, only this term remain. And so because we are more interested in the variance, as I mentioned, we are more interested in the variance of the parameters. So we want to calculate variance of these two phenotypic value. And look at here. Okay, so this is the locus to be tested, gamma. So because at this single locus, we have to estimate the IBD, the relationship between these two SIBs, right? For this locus. So we can use the data to calculate this, or if we do not have data, we can estimate this. But overall, for the polygenic, so that's the entire genomic, right? The entire set markers on the genome. So the variance of this polygenic effects between SIP1 and SIP2 will look like this. So because overall in the genome, so the SIP1 and the SIP2, they will share 1/2 of the same genetics from their parents, right? So the expected IBD value overall equals to 0.5. We have proved this, right? Right? So that's the whole genome. So that's easy to understand this. And this matrix is called the kinship matrix. Kinship matrix. So if you will do in the future, if you will do a lot of like GWAS analysis, you're gonna hear about these terms very often. So this means like you use this matrix structure to capture the relationship between the individuals in your study population. All right. So this is overall genetic variance. Okay, so this is the only difference. So other than that, you can use the same strategy to get the solution. Right. So you can construct the likelihood function, and then just follow the standard strategy to get the results. So it's very easy. So I'm almost done with this chapter. So I don't want to go deeper. So if you are interested in this part, so please read the textbook. And even we're done with this class. So, but if you are still interested in exploring more, so I will be very happy to help. So if you have in the future, when you have further questions, let me know so we can discuss together. So far, do you have any questions about this random model for QTL mapping?
So we use these so we calculate these the mixed linear or the linear model to
random model. Yeah, random linear model. Yes, here
random model, the random linear model, to calculate these we calculate these polygenic effects.
Yep.
And basically but we just talked about how we're using the variance instead of calculating the fixed model all the time, right?
Say that again.
You talked about how we use the variance to get these values instead of calculating the difference.
That's right. Because so if here we have too many alleles to consider, then the fixed model is not proper in this situation. Then instead of estimating the value for the allelic effects, we want to calculate this, the variance of the genetic effects. So if this is large, that means, okay, that means this locus is a impacting locus for the phenotypic value. If this variance is very small, that means that, there's no, so this variance captures the difference between the different allelic effects. This variance captures the variance of the different allelic effects, right? So if even at this locus, we're talking about a single locus, if there are multiple alleles, if all the alleles have the same identical effects, then the variance should be 0, right? Okay. However, if different alleles have quite different genetic effects, what do you see? You're going to see the variance of this different allelic effects will be subjected to a large variation. Do you agree with that?
Yes.
Okay, so this is the criteria we're going to use. The new criteria we're going to use for the random model to test whether a single locus is associated with the traits of interest.
Okay.
Does that make sense?
Yes. Now, how does the likelihood function put into this? You take
this is too complicated. That's why I don't want to go deeper. So the random model is a little bit different from it's less intuitive than the fixed model. All right. So so I just copy and paste this. So there are a lot of like very difficult, complicated theorem behind. I don't think we have time to discuss this for now. But you are interested. If you are interested in that, so we can talk about this later. How to construct a likelihood function for the random linear model, and then after you successfully set up this likelihood function, you use the same strategy to do the first derivative and the second derivative. So here the parameter is the variance of gamma, not the gamma itself, right?
Okay.
And then you can get the solution for the variance of the gamma. That's the MLE. And then you can either use the EM algorithm or if you have like missing data, the same strategy.
All right.
Mm-hmm. So yeah, here, this is too complicated. So I don't think I have time, because this VJ is something associated with here. Yeah, and this is the matrix calculation, and yeah, a lot of things. We need to derive here, but yeah, I think, yeah, I don't think we have time to do this now. But yeah, let's talk about this later.
Move on.
Okay. Any other questions? Okay, so chapter 14 is mapping QTL for multiple traits. So multiple traits means like sometimes like, so here we talk about like correlated traits. If two traits are totally independent, so that there's no way, so I don't think there's, so it is not that useful to analyze them simultaneously. Usually when some traits are correlated with each other, for example, if we are if we want to identify the genomic loci that are responsible for the human height and the human weight. So these two might be correlated with each other, right? So we can put these two traits together in Y to do the QTL mapping. And then, so in this case, the purpose of this is to identify the loci that are associated with both traits, which means like, yeah, this genetic determinant so the genes in this region. So if we identify some loci like that are simultaneously associated with two correlated traits, that means the genes in that region or some genetic determinants in that region are contributing to both traits. So this can help you to understand, to explore the network, the genetic network between the multiple traits and the genomic loci. So in rice, for example, so the multiple traits could be like the yield for the rice, or the heading date, or the KGW, which is the kilo grain weight or something like that. So if those traits are kind of associated with each other, correlated, we can put them together to do the QTL mapping simultaneously. So for example, so you can put different traits together, use the same strategy to do the mapping and likelihood function, and the rest of the analysis are very similar to what we have learned before. And this is the output, the Manhattan plot. If you analyze multiple correlated traits simultaneously. So here, different curve with different color represent. So each curve with a unique color represent a trait. So we have multiple traits here. So for example, in this locus, as you can see, many traits are, so many traits are or so it seems like this locus is associated with more than one trait, right? So the same thing here and here. All right, so this analysis can help you to dig in the genetics for all these traits, and can help you to understand the network, biological network behind these traits and the genomic determinants. All right. Any questions? Yeah, this is another example. Okay, so Bayesian, chapter 15. Bayesian, so this actually the same. So although we use a different term here, so because in the middle of the process, we use some Bayesian theorem to help us to get the solution. So the starting regression model is the same. So we use the same model, like here, mu plus A plus gamma plus, right? So the A here, we use A to represent the polygenic effects, and the gamma is the genetic effects for a single locus that we want to test. All right. So the same thing here. If we use this linear regression model, and we can get the MLE for any parameters of interest. And once we get this, we can calculate the posterior probability, like here, the posterior probability. Oops, here, sorry. We can calculate the posterior probability given the data. All right. So here we need to know the prior probability first, and then based on that, we can assume this parameter of interest is coming from a specific probability distribution. And once we can estimate the posterior probability for any parameter of interest, so we can do the simulation from sampling. So we usually simulate or sample any, so we do the simulation through the sampling. We can get the realized value for any parameter using this sampling. And the posterior sample will help us to get the estimation, accurate estimation for any parameter of interest. So this is very complicated, and it is not required for the final exam. And it takes time to explain this very clearly. So if you are interested in this, so please read the textbook, and yeah, if you have questions, yeah, we can discuss this in the future. And the empirical Bayesian QTL mapping is a modified version of the Bayesian QTL mapping. The only difference is like the Bayesian, the empirical Bayesian method is still a Bayesian method. The only difference is like, so in the regular Bayesian method, the hyperparameter, which is the parameter of the prior distribution, will be pre-selected by user. So for any parameters, you assume this parameter is following a prior distribution, and the parameters that determine, that control that prior distribution will be pre-selected by you. So you have the control on that. However, for the empirical Bayesian method, so you do not have that control. So you will estimate that hyperparameter, the parameters of the prior distribution from your data as well. So this is the only difference. All right. All right, now the further application, the chapter 17 and on. So we're gonna first introduce the microarray data analysis. So any questions so far before we move forward? Okay, good. So probably 10 years ago, the microarray, the gene chip are only used for measuring the gene expression profiles, the transcriptome profiles. But now we have developed a different forms of the microarrays, right? We also have the genotyping microarray, so that can help us to do the genotyping as well. But here, so we focus on the gene expression chip. So we're gonna see how we can use gene expression chip to get the profiles for the transcriptome in different sample, and we compare these different samples to identify the differentially expressed genes, right? So this is a very simple example here. Suppose we have two groups. The group 1 is treated by the treatment A, and group 2 is treated by the treatment B. Or you can think about like two groups, case control. The control is the normal sample, and the case means the treatment or the diseased sample, something like that. All right. So in each group, we have several replicates, biological replicates, because we need the replicates, right? Otherwise, the data is not trustable. So if we want to use statistics to analyze your data and get some reasonable conclusion, so we have to plan the replicates for each condition. So here, suppose we have a minimum of 3 biological replicates. So in each group, we have 3 mice, and from each mice we get the sample, so liver sample. So here, and we prepare for the sample and get the transcriptome sample and do the hybridization for each chip to get the data. So this is for the treatment A, and treatment B, we do the same thing to get the data for each single mouse. So this is the mechanism for the biological mechanism for the microarray. So microarray is a very small, tiny gene chip. So in this area, because it's very small, but it's very well designed. So if you zoom in, so you're gonna see on this chip, there are a lot of the chains, the DNA, the DNA chains. So we call these chains are the probes are attached on the surface of the array, right? We call these probes because this represented the entire transcriptome for either for the human or for the mouse. And okay, so during the hybridization, so we first label each sample with some specific fluorescence markers labels here, and we run through the gene chip, the gene expression chip analysis. We allow the hybridization occurring during this step. So if some genes are expressed, then the labeled target, the labeled the messenger RNA in the sample will be hybridized. Will match the probe on the surface of the chip, right? They're gonna hybridize and they're gonna stay. So if this gene is expressed, so the messenger RNA that represent this gene will stay on the chip, and then you're gonna see the signal, the fluorescent signal from here. So if this gene is expressed, you're gonna see this area which represent this gene will light up on this chip. So this is the very basic mechanism behind the gene chip, right? So this is the example showing, I mean, this is the old, very old data, because this is the data when I was a PhD student. When I was a PhD student, I analyzed the data like this. So this is from the Affymetrix. So in this table, you're gonna see each row represent a probe set, a transcript using different names here. And okay, so each column represent a single sample, right? And those are the values, the raw values from the gene chip. So this is a chip. And there are many probes here. And for each probe, you're gonna get a intensity value, right? So the fluorescence intensity value. So the higher means the more abundance for this transcripts. And okay, so this is the raw data. So if you just look at a single sample here, for example, the entire transcriptome, and if you plot this data, so this data usually looks like this. So let's see, 0. And this is very big numbers, suppose. So if you just plot the raw data for any sample, it will look like this. Most of the genes are very low expressed, and only So highly skewed data like this. So if you do not do any data processing, if you do not do any data normalization, if you just use the raw data, raw intensity data to do the data analysis, this highly skewed data will give you very unbiased conclusion. So usually we're gonna do what? We're gonna do the data transformation or the data normalization. So first, the data transformation. So we're going to transform data from this form. So if you see the original data is very highly secured. So after the transformation, we're going to get a data much nicely distributed, like the bell-shaped, so normal distribution. So this is the data after the transformation. If you plot that, so this will look like this. So there are many ways to do the data transformation. So there's a series of like Box-Cox transformation method. The most commonly used is the log transformation, because so the log transformation is like if your data is like this, you have something very extremely large data. So you use a log transformation to pull them towards the main body of your data. Then you're gonna make the data look nicely following this normal distribution, bell-shaped normal distribution. Okay. And the data normalization. Okay, so if you do not do the data normalization, you're gonna see. So if you compare different sample, so this represent the x-axis represent the different sample. So if you do not do the normalization, before normalization, you're gonna see different sample, the range, the distribution and the range of different sample are quite different. The variation is large. So this is not good because different chip, different batch may have different like systematic difference, the batch effect or the array effect. So we have to remove those unwanted effects before we do further biological analysis, right? So we have to normalize this data. So before the normalization, if you plot the MA plot, so this is a very special plot, MA plot, the data will look like this. What is MA? So for each gene, you have the average for the control group, right? And you have the average for the treatment group. So the A means if you add up these two average, the control group and the treatment group, that gives you the A addition. If you calculate the difference between C and T, the minus, then that gives you the M. So if you plot the M and A value, so this will give you this plot. Okay, so before the normalization, you're gonna see these points are not symmetric, which means when the gene expression is very low, you may have systematic bias or difference errors here. So we need to do the normalization. After we're done with the normalization, you compare this different sample again, so they are nicely in the same scale, so which is good. So this means like we have successfully removed some systematic errors bias before we can analyze the data to get more reasonable biologically sound conclusion. And after the normalization, if you plot your normalized data again using the A plot, so they are very nicely distributed, right? Everything is symmetric around this zero line. So any questions here? Okay, good. All right, so in the textbook, Dr. Xu mentioned many different ways to normalize the data, and this is the way he presented in the textbook. We can use a linear model to do the normalization. So for each gene in each sample, so this is the gene I in J sample and the case replicate. And we can use this linear model to model this observed gene expression intensity function or gene normalized expression function. Sorry, we can use this linear model to express the normalized observed gene expression value, right? So this is the intercept mean. So this is the overall mean. So we are not interested in that. This is probably the batch effect, the array effect, or something like that. And this is the genetic information we want to estimate the real difference we want to detect. So all right, so if we can use this model to estimate this, if we can use this linear model to describe the observed gene expression value, and then we can estimate this unwanted factors, right? Then what we can do is to do the normalization. We just remove this unwanted effect from the observed gene expression. And the remaining part is the normalized gene expression value. So by doing this, we can correct the observed gene expression value by remove the estimated unwanted effect. So the remaining part is reflect the real gene expression for this gene in this sample for this replicate. So this is one way we can do the data normalization. Any questions? Okay. All right. So once we are done with the data transformation and the data normalization, so we have cleaned the data, right? So we have improved the data for the analysis. Now we can compare these two groups. So we have A group, we have B group. Each one has 3 replicates. And for this gene, let's say for this gene, we have 3 values here and 3 values here. So we want to see if these genes are differentially expressed between A and B. So this is one test, right? So this is what we can do. So we can test whether mu1 equals to mu2. This is the null hypothesis. So we can test this. If this is not true, then we're going to take the alternative hypothesis, which means this gene is differentially expressed between A and B, right? So how can we do that? So this is the 2-sample t-test. So let me give you an example. So suppose you want to compare farm A and farm B to see which farm is sweeter, which means like which farm can produce sweeter fruits. All right. So what you can do is like you just randomly pick some fruits, a basket of fruits from farm A and farm B, right? So the random is very important. If you do not do the random sampling, so then that will give you biased results. If you do this random sampling, that means like the sample you get from each farm will represent the fruits generally, right? You overall will represent the overall fruit quality from that farm. All right. Now, if you have 2 baskets, one from each farm, so how can we compare the overall fruit quality from these 2 farms? Simple. You just put them together, squeeze them together, mix them together to generate two glasses of the juice, right? And you taste it. So to compare this, the average of these 2 baskets to see which one is sweeter. So immediately you can get the results. So this is what we do here. So we do the t-test. We compare two group means to see if they are identical or not. So the simple way to do this is to do the 2-sample t-test, and following this is the degree of freedom, and following the routine process, we can get the p-value, and the p-value can tell you whether this gene is differentially expressed or not between group A and group B, right? All right. So now, as you can see, we are not just satisfied by testing one gene. We have to test many genes in the entire transcriptome, and this involves the multiple comparison. So we have talked about these concepts before, right? So when you test many hypotheses simultaneously, so this will so if you do not adjust the p-value, this will increase your family-wise or experiment-wise type 1 error. So we have to do some adjustment or control on the forced discovery. So one way we can do is the Bufferoni. And the Bufferoni is like, so for the regular 0.05 cutoff, so we can divide this 0.05 by the number of tests and give you a new significance level of to be used for each individual test. But sometimes, yeah, this is too conservative. So then we can choose to use some other strategy, either the simulation permutation. Hopefully you still remember the permutation strategy can help us to generate a little bit relaxed p-value for your multiple tests. And you can also use the BH, Benjamin, I forgot the other name, but BH FDR process to help you to generate a relaxed cutoff for your multiple tests. Okay, so once you analyze your data, so this is a example, the sample output from your analysis. So all the genes are listed here, and this is the gene title. So telling you what the gene is for, what the gene is doing in the cell, and this is the gene's symbol, and the log folder change. So the positive means the increase, the upregulated, and then the negative means the downregulated. And this is the T statistic. So this is the average value for the gene, yeah, overall average value, and give you the p-value, original p-value here, and the adjusted p-value. So this is the adjusted p-value. And also, so the package I use is called the NEMA, linear model analysis for the array data. And this method will also give you a b-value. This b-value is the posterior is a statistic generated from the posterior probability. And usually when the genes has the b-value greater than 0, then that means this gene is differentially expressed between these two groups. All right. So right now, as most of you have already known, so still like microarray has been widely used currently, but we have developed very many new platform for measuring the gene expression profiles. For example, we can use the RNA-seq method to analyze the data. But from that platform, the data you got for single gene is quite different. So from this RNA-seq, you're going to get the sequence data, and the output for that sequence data is the count number of each gene. So for example, in your sample, if you analyze your sample using the RNA-seq for a specific gene, you can get maybe 100 counts, which means in your sample, that gene has been represented 100 times in the sequence in your sequence data. So of course, the count data is not following a normal distribution. So usually the count data is following a Poisson distribution. So many bioinformatics tools have been developed to analyze those kind of data. The model that is commonly used to analyze the count data from the RNA-seq technology is the negative binomial distribution. So many statistical methods have been developed to use that model to analyze the RNA-seq data. However, as I mentioned, so if we can transform the data into a different distribution, we can still use the method that we have developed for the analysis of the microarray data to analyze your RNA-seq data. So they are the same actually. Once you can successfully transform your count data into the normally distributed data, you can still use the existing microarray methods to analyze your sequence data. All right. So here, I just give you an example showing how you can identify the differentially expressed genes between two groups, A, B, or the control and the treatment, right? The case control. So if you want to test more than two groups, if you want to identify the differentially expressed genes from more than two groups, then you can use ANOVA or F-test to help you. So we briefly described this before, right? So this is an extension of the t-test. T-test is good for two sample analysis. The F-test is good for more than two samples, more than two levels, right? And so once you get the microarray data, gene expression profiles, transcriptome data, so you can do a lot of interesting analysis. So for example, you can see whether genes will change, the gene expression profile will change along the treatment time. So if you treat the individual and you get the data at different time point, so you can see if any gene is responsible, well, if you, so by doing this, you can see if a single gene respond to the treatment, right? So if along the time, if you majority of the genes will not change, so they fall in this group, no change group. But for some genes, they show the same trend. So they increase at the beginning, they decrease at the beginning, but increase as time goes on. And some will consistently increase, and some will show this S shape, something like this. So by doing the clustering, you can identify the different group of genes that showing different pattern along the treatment time, and this can give you insight into the network about the disease or about the treatment itself. Also, you can treat the gene expression as a quantitative trait. So yeah, this is so gene expression is not the traditional traits, but still because they are varying in a quantitative way, right? So you can treat each gene as a quantitative trait, and you can find the genes that are associated with the traditional traits. For example, the disease traits, disease phenotype, or in a rice, for example, the traditional traits, yield. So you're going to see which gene is associated with the traditional traits and find the pattern of the association between them. And this can also gives you insight into the genetic pathway or genetic network. There's another very famous study called the eQTL mapping, because QTL mapping is very easy, right? So we have been talking about this a lot of for a lot of times. The QTL mapping means like we try to find the genomic loci that are responsible for the traditional traits. The eQTL means, yeah, this is not the traditional traits anymore. The E means the expression traits, which means we can treat each gene, the expression profiles for each gene as a quantitative traits, and we can map each gene into the genomic loci. So as you can imagine, right, for most of the genes, if there's a variation, so they are more likely to be located in cis. So in cis. So the gene change itself will, so the mutation in a gene or there's something happened in that gene will change itself change the abundance of that gene itself. So that's the cis mapping. And because some genes, if there's some change in one gene, and this gene can affect the gene expression abundance for another gene in another location in the genome. So that's the trans mapping, and this can also gives you the insight into the network biological pathway of different genes and the genomic loci. So for example, this is a typical output from the EQTL mapping. So this is the genome. And as you can see at this locus, a lot of transcripts are mapped here, and a lot of transcripts are mapped here, which means like this locus is very important. It may control many genomic transcripts in a sample. All right, I think, yeah, that's all. So congratulations to, to all of you. All right, we're done with this difficult class. I hope you have learned a lot from this class. So any questions for today's lecture? So I know, yeah, we moved forward very quickly. So yeah, you definitely you have some questions for the stuff we have covered today. So any questions?
Mm.
Any questions for today's lecture? Good. Yeah. Hi. Yeah.
Right.
Go ahead, Ryan.
So you talked about how the gene expression of the phenotype was, you're talking about how the gene expression, the gene expression traits related to traditional traits. What did you mean by traditional traits in this case?
Traditional traits, for example, so let me use a human disease as an example. So suppose you have a group of patients, and each patient have some traditional traits. For example, the disease status, the Gleason scores, or the aggressiveness of the tumor, right? And the easy way for you to identify the genes that are contributing to this trait is to get the correlation between the traits and the gene expression. Do you agree?
Mm-hmm.
So you, sorry, excuse me. So so before you can do many like different kind of advanced statistical analysis to find the association between gene expression and the phenotype. The simplest way, the intuitive, the easiest way for you to identify the potential genes for this disease is to do the correlation, right? You correlate each gene expression profiles with these traits. The traits is quantitatively varying, right? So if you see the pattern, okay, yeah. But usually this is not the good. This is not the best strategy. So why? The same reason. So why we need to include the polygenic effects into the regression model to do the test, because if you just test the single gene with the traits, you just ignore the contribution from other genes, right? In your test, that may not reflect the real situation. That may not give you a enough power to detect the association between this gene and the traits. So what we can do is like we can leverage more advanced statistical analysis to include this gene and many other genes as the background to test if this gene in that background is still associated with the traits of interest, right? Yeah. So, but the easiest way, the straightforward way is to do the single correlation analysis. This can help you to identify very, very strong genes. So if this gene is really strongly associated with the traits, yeah, you can identify it immediately, right? But for some like mediocre, like like the genes, still they are very interesting and they are very important. But by just looking at the simple correlation between that gene and the traits, you can never, you will never be able to identify pick up the association. So in this case, so the statistician will play a very important role. So you're going to use advanced method to pick up the association, even in the very noisy background. Does that make sense?
Yes.
Mm-hmm. So any other questions? Very good. So again, so I'm going to remind you, so tonight, 11:59, please, please submit your term paper. And and Thursday morning, so, I'm looking forward to your presentations. Yeah. All right, thank you. Bye-bye for now. Bye.